While we are aware that stem cells have an alternative genome to the patient's own genome, we have no evidence at this point. In fact, we have evidence to the contrary. No evidence that there is any engraftment of that foreign genome into the patient. And in fact, we've had many thousands of administrations of the product with no evidence of a foreign genome representing itself or being reflected later downstream in the patient care.
Does that then portray a situation where someone receives a procedure with postnatal stem cells, and then it's really more of their own body harnessing its own regenerative capability as a reaction to those stem cells being applied?
That would be my hypothesis at this point, based on all the data that we have. I do believe that the patient is benefiting from the cells in a way that's non-engrafting, where the genome is not actually being overtaken by a foreign genome. In fact, we believe that the cells are secreting growth factors and various molecules that will help draw that patient's own machinery, regenerative machinery, to the wound or to the area that needs regeneration.
What is the difference and effectiveness between using, let's say, postnatal stem cells versus the autologous options that are out there?
The way our bodies work throughout our entire lifespan, including from the time we're newborns to and even gestating to the time that we're on our deathbeds, is that we regenerate and heal ourselves. In fact, we all know that multiple parts of our bodies and tissues and organs are completely replaced multiple times in our lifespan. So we know that these things that we're using, whether it's bone marrow or adipose or PRP, are things that are already present in our physiology. And so the question I have, and the question many have, and our current understanding is, if those things aren't already functioning properly in the patient, then why would concentrating and re-injecting them into certain spots be a more effective treatment than a graft? A graft that would come from a live, donated, postnatal contribution. These things are compelling in their ability to potentially overcome the deficiencies that are in this patient, which is the reason they're actually seeking medical care in the first place.
One question a lot of doctors ask us is, these cells that are being administered, let's say into the knee, do we have research that shows how long the longevity is inside the knee?
Great question. And in fact, there's a really interesting study done by a group that I've actually corresponded with. I think it was maybe 5-6 years ago. I don't do well with how long ago something happened. The principal investigator is in Australia. They did an interesting experiment where they took human umbilical cord tissue cells and labeled them with ferraheme, or ferromoxetol, which is something that can show up in an MRI. They incubated it with this molecule that was not toxic relatively, and then did an IV into pigs whose hearts had been artificially injured. They then were able to track these pigs in an MRI. Don't ask me how you get a pig to hold still in an MRI, or where to give a pig an IV between its hooves. But they then tracked these cells in the pigs and where they were located using an MRI. And they found that 6 weeks later, so this is a significant amount of time, and remember here we're talking about human cells that have been artificially engineered to register on an MRI. 6 weeks later, the cells had all aggregated into the wound in the pig's heart, artificially induced wound. Certain length, certain incision, and those pigs who'd been administered the labeled cells, hearts had healed.
And the cells were still present at the 6-week
Correct.
time
6
frame.
weeks, alive and present.
I know that the processing of the postnatal donation is federally regulated heavily, and within that, you have been able to improve those processes. Can you address that? I don't need you to go into the actual, intricacies, but I also want you to address the cryogenic freezing and how long can it stay like that?
Great questions. The FDA regulates our space rigorously. The primary objective is to minimally manipulate the postnatal donation. And so there are requirements there. Within those requirements and within that framework, we've been able to make huge improvements incrementally over a period of many years, to the point where we're able to retrieve a more viable, more regenerative sample post-thaw. Once we cryopreserve it, the sample itself can stay in cryo, and this is at temperatures of liquid nitrogen, which are minus 384 degrees Fahrenheit, I believe, or 192 Celsius. It can stay at that temperature in liquid nitrogen for many, many years and still be just as viable after a decade as it was after a year of such storage. And in fact, we have our own, because our company is 15 years old, we have our own data that confirm that awesome result. We minimally manipulate this postnatal donation. We do not gamma irradiate. We do not add enzymes. We do not add any exogenous molecules or reagents that could potentially take it outside of the scope of minimally manipulated. It's as natural as it can possibly be. And part of the reason that the viability of cells is what it is post-thaw is because of that process, the way we do it, how quickly we do it, and the fact that we have control over every single part of the process.
Follow-up question to that. My understanding, underneath those minimal manipulation guidelines, is that there are some variations to where you can create proprietary processes that make the biologics purer, more lively, energetic with the cell activity. Can you speak to that?
Absolutely. It's really an interesting puzzle to solve. And having the live cells is an indication of how that whole process was done from start to finish, not even just the donation. So it's easier to talk about what we do than it is to talk about what we don't do to keep our process and the product as pure as possible. While we do have proprietary portions of the protocol, the purity of the process and the product mean that we don't use gamma irradiation, which destroys live cells. We do not add reagents or enzymes to break the product free of its extracellular matrix. And so in the end, the product itself is pure and as native to the patient's physiology as their own cells would be, as opposed to even just a molecule that you go buy off the shelf at Walgreens, which is a foreign molecule. Let's take ibuprofen, for example. That is something that is not native to our bodies. But every single component and part of what is in our product is something that our body's already seen and is actually seeing on a regular basis. It's just young, vibrant, healthy, and regenerative.
From a science perspective, when a patient receives the postnatal stem cells, what is happening with regards to amping up your body's capability to regenerate, as opposed to what it would normally do without that contribution in a procedure?
The graft here is the key. The graft is actually bringing in resources and capabilities from an external source into a patient who is potentially older and less capable of auto regeneration, which happens to all of us as we age. That is really sort of the miracle that is presented when you're offering something that, and I'll list off a few key differentiators between the cells that you would get from your own body and the cells that you would get from an allograft from postnatal tissue. So the fact that we're calling it an allograft is interesting. Graft is the key word, and grafting is essentially what we're doing when we're taking a postnatal donation and bringing it into a patient whose auto-regenerative capabilities are diminished, as is the case as we age. The differences, the key differences between a patient's own stem cells and the grafted cells that come from postnatal donations are, number one, the secretome or the exudate. The things that are secreted by these cells are very different. They change as we age. They change with the cell type. But certainly, creating an entire human being over a period of 9 months with all of the support tissue that makes that possible, the umbilical cord, the amniotic membrane, the placenta, the cord blood, all those things that are left behind and would otherwise be discarded, those things host an amazing machinery, which includes cells that are secreting at a much higher rate regenerative and growth factors, non-inflammatory but growth factors, non-carcinogenic but growth factors that actually will have been able to create an organism in a short period of time. In addition to that, the number of divisions that remain in a cell from the perinatal birth donation is huge in comparison, orders of magnitude more than the cells that we have in our own bodies, whether it's our bone marrow or adipose. So that's a huge difference as well.
So, Dr. Spencer, I wanted to ask you about the process for procurement and how the postnatal donations are processed, because I know it can be a very intricate scenario.
What's exciting about the way we procure our healthy donations now is what is possible with the live cell count and the health and the regenerative potential of these things because of the way we're doing it and the fact that we have the ability to be in control of every step in the process, which is unique to us. As far as I know, we are unique in the ability to be able to control from the very moment that the doctor proposes to the mother that there's a potential use for the donation, besides having it discarded, to the point where we cryopreserve it and then thaw it out and have a live cell count that is remarkable. And frankly, compared to what was possible 10 years ago, honestly, it would have been unbelievable at the time. So that's what is so unique about what we do, and it's why we try to do every step of the process here from start to finish.
So I think it's important to recap that you basically control the whole process as far as working with the OB offices, the moms themselves, the hospitals, and nothing is outsourced. Medical processes and things advance over the years, and this is no different. So can you speak to some of those differences and how you're able to achieve a better result with those donated, after those donated procedures?
Yeah, great question. The first thing that was necessary was, just as you said, us as a group, a company, taking over every step in the process. And until we were able to do that, we weren't able to make the tiny improvements that we've made over the last 10 years in every facet of every part of the process. And it's multifaceted. From the time that our own people go and pick up the postnatal donation at the time of the procedure from a local hospital with its short driving distance from our laboratory, to the way that we package it and we instruct the doctors, not only with how they screen the donors, because it's actually very rigorous selection process. We don't accept donations from just anyone, to the point that we're actually immediately processing, and the way we're processing it and the steps of the processing, the reagents that we're using, the methods that we use to cryopreserve the product have improved in the last 5 years alone, that have been able to increase and improve our cell viability between 10 and 15% over the last 10 years from where they were 10 years ago. Those being able to be involved and have our hands on every step in that process have made a huge difference in the final outcome.
When you look at the United States, we use first-generation cells that come from the postnatal donation. Can you speak to the benefits of that as opposed to culturing, which we don't do in the United States, that could then go down second, third, 15th generation? I'm just curious to hear your thoughts on that.
That's a great question. In our country, in our current regulatory environment, we are not allowed to expand or culture the stem cells, which initially, at first glance, sounds like a really fun and exciting thing to do. You can grow a stem cell farm. But the reality is, with every single generation of cell division and expansion, as exciting as it sounds to start with a few cells and end up with millions of cells, and having done this myself in a laboratory, I've been in a lab for the last 30 years of my life, probably, something like that. And it is true that you can put cells in culture, feed them the right things, and get them to grow, and end up with 1 billion cells when you started with 100,000. The reality, though, is that at the end of that expansion or cell culture, you have a very different cell, a very different cell population than you did at the very beginning. Those cells that were at the beginning were capable of doing that, but doing it in vitro instead of in vivo in our bodies, in our knee, our shoulder, wherever, whatever we're treating, is we still are not fully we do not fully understand what that final cell is going to be, and we can't aim it. And so by the time we harvest those expanded cells, they have a variety of characteristics that are very different from the initial cells that were put into the culture. One of those is how many divisions they have left. As simple as telomeres are, and as perhaps reductionist of the idea of telomeres is, our cells do in the genome. Every time we divide a cell, there has to be a copy made of that cell's genome or DNA. As that gets copied and copied and copied and copied, that actually reduces the number of future copies possible. That's one difference. The other differences are things that just start to fall apart. They just enzymes and molecules and transfer RNA and so on. The cell is not able to actually continually maintain that perfect, pristine initial state. And until cell culture advances to the point where we can actually do that, those cells, I would prefer not to use them myself.
So we know that stem cells have DNA. So one of the questions I want to ask you is, when you have a stem cell procedure with postnatal biologic, how does that play out in the procedure with the patient?
So the way the actual genome reflects in the therapy is at this point relatively unknown. But we do have evidence that suggests that there is no engraftment taking place. So, and that was interesting. We were imagining that perhaps that there would be engraftment, that we would have cells that would actually help to regenerate the wounded area or the part of the body that needed regeneration. But at this point, we're finding that they're actually acting more like nanobots, let's say, and that they're secreting growth factors and various things that will actually recruit that patient's own machinery to the site. And that in the end, any new part of the body that's regenerated is actually that patient's own genome. And so we're not seeing foreign DNA yet. There are some interesting experiments that we would like to do, biopsies where we do genetic testing. Some of those things have been started, but I don't think we're there yet.
When you look at the United States, we use first-generation cells that come from the postnatal donation. Can you speak to the benefits of that as opposed to culturing, which we don't do in the United States, that could then go down second, third, 15th generation? I'm just curious to hear your thoughts on that.
That's a great question. In our country, in our current regulatory environment, we are not allowed to expand or culture the stem cells, which initially, at first glance, sounds like a really fun and exciting thing to do. You can grow a stem cell farm. But the reality is, with every single generation of cell division and expansion, as exciting as it sounds to start with a few cells and end up with millions of cells, and having done this myself in a laboratory, I've been in a lab for the last 30 years of my life, probably, something like that. And it is true that you can put cells in culture, feed them the right things, and get them to grow, and end up with 1 billion cells when you started with 100,000. The reality, though, is that at the end of that expansion or cell culture, you have a very different cell, a very different cell population than you did at the very beginning. Those cells that were at the beginning were capable of doing that, but doing it in vitro instead of in vivo in our bodies, in our knee, our shoulder, wherever, whatever we're treating, is we still are not fully we do not fully understand what that final cell is going to be, and we can't aim it. And so by the time we harvest those expanded cells, they have a variety of characteristics that are very different from the initial cells that were put into the culture. One of those is how many divisions they have left. As simple as telomeres are, and as perhaps reductionist the idea of telomeres is, our cells do in the genome. Every time we divide a cell, there has to be a copy made of that cell's genome or DNA. As that gets copied and copied and copied and copied, that actually reduces the number of future copies possible. That's one difference. The other differences are things that just start to fall apart. They just enzymes and molecules and transfer RNA and so on. The cell is not able to actually continually maintain that perfect, pristine initial state. And until cell culture advances to the point where we can actually do that, those cells, I would prefer not to use them myself.